Translation. We have studied that DNA, by the process of transcription, forms mRNA, that is messenger RNA, and that actually carries the code for the synthesis of the protein in the form of genetic code. And that genetic code is then translated to produce the certain kind of proteins. We have also studied that the genetic code is a triplet codon, and that has to be again read so as to form a certain type of protein. Now, translation, when we say or define, translation is a process in which the genetic information is genetic information coded on mRNA is converted into polypeptide chain to form protein, as determined by the sequences of bases on mRNA. So that simply means that the DNA, which has passed on the information through the complementary bases to mRNA, is now converted to form the proteins. So this process is known as translation, because in this we are only decoding the information and synthesizing of the protein is taking place. Now here the role of RNA is very, very important. We know there are three types of RNA which are existing. That is, first is mRNA, then we have the tRNA, and then the rRNA. The mRNA is known as the messenger RNA because it is passing the information. It is carrying all the information and passing all this information, which is to be decoded to form the particular protein. And this mRNA forms around 3 to 5% of total cellular RNA. So the total cellular RNA, which is the total amount of RNA which is present in the cell, it forms only 3 to 5%. And it is found on specific parts of DNA, that is the structural gene. So mRNA is found only on the structural gene part, and its sequence is complementary to that of the DNA. The tRNA is a very important RNA, and it is formed from a small segment of DNA. The tRNA is also known as soluble RNA. It is also called as soluble RNA. Soluble RNA for the reason because it is soluble in 1 molar HCl, 1 NaCl. So therefore it is known as soluble RNA. What is its function? Its function is to carry the amino acids during the protein synthesis as per the mRNA. And it is a single stranded structure and is looped to form a clover leaf shape. Now when we say clover leaf, it has a specific size and specific shape. The clover leaf shape is like this, which has certain zones. If we say now it is clover leaf shape, and tRNA has an anticodon loop. This is known as anticodon loop. this lower part is known as anticodon loop. And when we see the polarity, it is 3' and 5' over here. This anticodon loop, it has bases, and these bases are complementary to the base or the code on the mRNA. So this anticodon loop will have, suppose this we are talking of as mRNA, this is the mRNA, then this site, that is the anticodon loop, it is the 3 codons on the mRNA, and there will be 3 complementary codon, that is anticodon loop, and it will be complementary to that to the mRNA. Also, it has amino acid acceptor site, to which it binds to the amino acids. Since the tRNA has anticodon loop, the sequence over here will be fixed, and it will also be complementary to the mRNA. Therefore, we say that the tRNA is specific for an amino acid. For example, if it has an anticodon over here as UCA, and on mRNA it will have AGU, because this, why we are using U, because it is RNA. So it will be, this is the information in the form of code which is in the mRNA during the transcription process. The mRNA is carrying this information, and the tRNA, which is having this information, will attach itself to this particular point. That is why it is known as anticodon site, and it is specific. If the sequence over here is UCG, then it will not bind over here with this mRNA, because now the sequence has changed. So it has to be UCA over here. When it codes for this particular is coding for SER, that is the genetic code. Now there are no stop codons over here. There are no stop codons in the tRNA. Also, the structure is a typical structure because it facilitates the binding or the attachment of this particular mRNA codon. It is a very compact molecule, and it is specific for each amino acid. Lastly, this is about the tRNA. When we talk of rRNA, that is the ribosomal RNA. Ribosomal RNA forms around 80% of the total RNA, and it is a component of the ribosome. Ribosome, we know it has 2 subunits, one larger unit and the one smaller unit. So the ribosomal unit is formed from the small segment of DNA, but there are no definite base relationship. It is the organelle that helps in the translation of mRNA. That is, tRNA is breaking up the code, and rRNA, ribosomal RNA, helps in the formation of the protein. So the complete RNA, that is mRNA, rRNA and tRNA, take part in the translation process.